Hello students, today we can see the topic Chemiosmotic Hypothesis. What is chemiosmosis? It is the movement of ions across a semi-permeable membrane-bound structure down their electrochemical gradient. Down their electrochemical gradient means from a region of high concentration to a region of low concentration across the semi-permeable membrane. ATP synthesis also occurs by chemiosmosis. In ATP synthesis, hydrogen ion will be moving across the thylakoid membrane from the lumen of the thylakoid membrane to the stroma of chloroplast. So we can say ATP synthesis occurs by chemiosmotic hypothesis. The chemiosmotic hypothesis has been put forward to explain the mechanism of ATP synthesis. This ATP synthesis is linked to the development of a proton gradient across the thylakoid membrane. What is a proton gradient? It is the difference in hydrogen ion across the membrane. The membrane here is the thylakoid membrane. ATP synthesis can occur in mitochondria as well as in chloroplast. So here we are seeing how ATP synthesis occurs in the chloroplast. Okay, so inside the chloroplast in the grana, thylakoid membrane is there, and this thylakoid membrane is having a lumen inside it. The membrane encloses a lumen in it. Lumen means the space or cavity which is enclosed by this thylakoid membrane. So inside this lumen, proton gets accumulated. Okay, so the hydrogen ions or protons will be getting accumulated in the lumen than the stroma. Stroma is the matrix of the chloroplast. Okay, so when more and more proton accumulates inside the lumen than the stroma, then the proton gradient will be created. How the proton is getting accumulated inside the lumen? One reason is due to splitting of water that occurs inside the thylakoid membrane, that is on the inner surface of the thylakoid membrane. The splitting of water occurs, which releases more amount of hydrogen ions. Okay. Then as electrons move through the photosystem during the non-cyclic photophosphorylation also, hydrogen ions will be transported into the lumen from the stroma. Okay, then due to reduction of NADP, when NADP is reduced to NADPH, hydrogen ion will be used up from the stroma. Okay, so hydrogen ion concentration decreases in the stroma and it gets increased in the lumen of the thylakoid membrane. Okay, thus a proton gradient will be created. For chemiosmosis to occur, it requires a membrane, that is the thylakoid membrane, a proton pump which is used to pump the protons from the stroma into the thylakoid lumen. Then a proton gradient should be created. Then ATP synthase enzyme which is used to synthesize ATP. Okay. So what are the requirements for chemiosmosis? Chemiosmosis requires a membrane, a proton pump, a proton gradient and ATP synthase enzyme. Now energy is used to pump the protons across the membrane. So when energy is used, the protons can be pumped from the stroma into the thylakoid lumen so that a proton gradient will be created across the thylakoid membrane. That is, proton will be accumulating more in the thylakoid lumen than the stroma. Thus, a proton gradient will be created. Now we can see how the proton gradient is created and ATP is synthesized. One step by which proton gradient is created is due to the splitting of water. Okay. The photolysis of water occurs in the inner side of the thylakoid membrane. So here this is the thylakoid membrane and this is the lumen of the thylakoid membrane and this is the stroma of the chloroplast. Okay. So the water splitting enzyme complex is present on the inner side of the thylakoid membrane in the photosystem 2. Okay. So when light is absorbed by this photosystem 2, here when light is absorbed by the photosystem 2, As a result, photolysis of water also occurs. Okay, so the enzyme complex which is responsible for splitting the water molecule into oxygen, hydrogen and electrons. Okay, so oxygen, that is oxygen atom, plus hydrogen ions and electrons are released as a result of splitting of water molecules, isn't it? So these electrons will be used up in the electron transport system. Okay, and the oxygen atom will be forming the oxygen molecule. That is, when two molecules of water is split, then oxygen molecule or oxygen gas is formed, which can be released out of the thylakoid membrane. It will be coming out of the thylakoid membrane and it can be released out as a oxygen gas, one of the byproduct of photosynthesis. Okay, then the hydrogen ion, it gets accumulated in the lumen. Why it gets accumulated in the lumen? Because it is released inside the lumen of the thylakoid membrane. Okay, so splitting of water leads to increase in hydrogen ion concentration in the lumen of thylakoid. Okay, so how it is happening? When the photosystem 2 is absorbing the light photons, the enzyme complex which is responsible for splitting the water is present on the inner side of the thylakoid membrane in the photosystem 2, and that will split the water molecule into hydrogen ions, electrons and oxygen atom. This oxygen atom will be forming oxygen gas and then it will be coming out of the thylakoid membrane and it will be released out as a byproduct of photosynthesis. The electrons will be used up in the electron transport system and the hydrogen ions will be released in the lumen. Thus, the hydrogen ion accumulates in the lumen of the thylakoid membrane, which is responsible for creating the proton gradient. Okay. Proton gradient is also created during the non-cyclic photophosphorylation when the electrons move through the photosystems. In non-cyclic photophosphorylation, the photosystem 2 will be receiving the light photons, isn't it? So when it receives the light photons, the reaction center, that is the chlorophyll A molecule, will receive that light energy and the electrons get excited. So when the electrons get excited, it will be moving farther away from the atomic nucleus. It moves to an orbit farther from the atomic nucleus, isn't it? And that electron will be picked up by the primary electron acceptor, which is present on the outer surface of the thylakoid membrane, that is in the photosystem 2. Okay. And from there it will be transported, that electron will be transported to the electron transport system, which consists of plastoquinone, cytochrome complex and plastocyanin. So the electron will be passing through this electron transport system and then it will be received by the photosystem 1 or the P700, isn't it? So now when the electron is passing through this electron carriers in the electron transport system, along with this electron, hydrogen ion will also be carried. Why? Because these are not actually electron carriers, they are hydrogen carriers or proton carriers. Okay, so when the plastoquinone is receiving the electron from the primary electron acceptor, it will also take up an hydrogen ion from the stroma. Why? Because plastoquinone is not actually an electron carrier, but it is a hydrogen carrier. So when it carries the electron, along with that it will be carrying a hydrogen also. Okay, so it carries the proton from the stroma. Okay, and then it passes the electron to the next electron carrier, that is the cytochrome complex. Okay, the cytochrome complex also will be receiving that hydrogen ion or the proton. Okay. And as you see, this cytochrome is towards the inner side of the thylakoid membrane, isn't it? So when it carries that hydrogen ion, it will be releasing that proton or the hydrogen ion into the thylakoid lumen. Okay, so while it is transporting the electron, along with that it carries the hydrogen ion also, and it releases the hydrogen ion into the lumen of the thylakoid membrane. Okay, and then it passes the electron to the next carrier, electron carrier, that is plastocyanin. Okay, so whenever the plastoquinone and the cytochrome is transporting the electron, actually they are not electron carriers, they are hydrogen carriers. So along with the electron, they will be transporting hydrogen ion also. So from where they transport the hydrogen ion? From the stroma. So along with the electron, they transport the hydrogen ion or the proton from the stroma and then release it into the lumen of the thylakoid membrane, and thus the lumen of the thylakoid membrane will be receiving more amount of protons. Okay, so the number of protons in the stroma gets reduced and number of protons in the lumen of thylakoid membrane gets increased, and thus a proton gradient will be created. During the non-cyclic photophosphorylation, the electrons which are released by the photosystem 2 will be passing through the electron transport system, and now it will be reaching the photosystem 1, isn't it? And that electrons as well as the electrons which are released by the photosystem 1 will be used up by NADP to get reduced to NADPH and H+. So for the reduction of NADP to NADPH, electron is used as well as hydrogen ion is also essential. Okay, and the enzyme which is used for the reduction of NADP to NADPH and H+ is NADP reductase enzyme, and it is located on the stroma side of the membrane. So this is the stroma side. So here what is present? The NADP reductase enzyme is present. So this NADP reductase enzyme, as it is present towards the stroma side of the membrane, it will be utilizing the electron as well as the hydrogen ion from the stroma to reduce NADP to NADPH and hydrogen ion. Okay, the electron which is received from the photosystem 1 will be utilized for the reduction of NADP to NADPH. Along with that, what is also required for the reduction of NADP? Hydrogen ion is required. So from where the hydrogen ion is used up? From the stroma. So this is the stroma, isn't it? So from the stroma, the hydrogen ion is used for the reduction of NADP to NADPH and H+. So what happens? The hydrogen ion concentration present in the stroma will be decreasing. Okay. Now we can see how ATP is synthesized. So for the synthesis of ATP, a proton gradient is required, and now the proton gradient is created. You can see more number of protons will be accumulated in the lumen of the thylakoid membrane than the stroma, isn't it? Now this gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. How the gradient is broken down? The gradient is broken due to the movement of the protons across the membrane to the stroma. How it is moving? It will be moving through the transmembrane channel. So here certain proteins will act as the transmembrane channel through which the protons will be moving from the lumen into the stroma. Okay, here the ATP synthase enzyme will be acting as a transmembrane channel. The ATP synthase enzyme consists of two parts. One part is called the CF0. So this is the CF0 part. What is that CF0? C means channel, F0 is fraction 0. as I told, ATP synthase is having two parts, isn't it? One fraction is CF0 and another part is what? CF1, that is channel F1 or fraction 1 of ATP synthase. Okay, so this CF0 part of the ATP synthase forms the transmembrane channel as it is present within the membrane, within the thylakoid membrane. So this is the thylakoid membrane, and within that membrane this CF0 part is present. Okay, and the CF1 part will be protruding, or it will be projecting out of the membrane, thylakoid membrane. So it is seen towards the stroma side. It will be facing the stroma side. Okay. So the CF0 part is present within the thylakoid membrane, which acts as the channel through which the hydrogen ions will be passing from the lumen of the thylakoid to the stroma. Okay, so it will be passing like this. Okay. So, when the hydrogen ions will be passing through this channel, that is through this CF0, and when it reaches the CF1, what happens? Some energy will be released. Okay, when the hydrogen ion is moving from the lumen to the stroma, some amount of energy will be released, and that energy will lead to some change, conformational change in the CF1 particle of the ATP synthase enzyme. Okay, so when such changes occurs in that enzyme, what happens? It helps in the reaction of ADP with the inorganic phosphate. Okay, so it helps in the combination of ADP, that is adenosine diphosphate, with the inorganic phosphate. As a result, ATP will be synthesized. Okay. So the conformational change that takes place in ATPase enzyme, that is in the CF1 particle of ATPase enzyme, will help the ADP to combine with the inorganic phosphate. As a result, ATP will be synthesized. Thus, ATP synthesis occurs in the chloroplast. Okay. Thus, the ATP which is synthesized by chemiosmosis and the NADPH which is produced by non-cyclic photophosphorylation are used immediately in the biosynthetic phase. The biosynthetic phase takes place in the stroma. Okay, and during the biosynthetic phase, the carbon dioxide will be fixed and carbohydrate or sugar will be synthesized. So the end product of light reaction, that is ATP and NADPH, are immediately used in the biosynthetic phase, or the dark reaction, during which carbon dioxide will be reduced to carbohydrate. Okay.